idea. So this morning I came in and passaged those cells. So who wants to tell me the steps I would have done? I checked them out of the incubator. They all looked good. There was no contamination. I checked for a thing called confluency. Does someone want to raise their hand and tell me what confluency is, kind of? Yeah? So confluency is a description of how much of the cells are covering the dish. So if they're over confluent, they're too crowded. And if there's not a lot of them, they might be under confluent. And so usually you want them to be, it depends. Some cells like to be really crowded, like cancer cells, some cells don't. So because we just passaged them on Thursday, they weren't super crowded, so I just combined all of those dishes together to get enough for our slides. So after I looked at them, what would I do? I'd take out the media, right? And then what? Rinse them with PBS, which is my salt buffer. And then what do we do? How do I get them off that dish? Because they're adherent cells, right? So they stuck to the bottom of the dish. Yes? Yeah, you have to attach them using an enzyme. Does anyone know the name of that enzyme? Yes. Trypsin, yes. And triply is a less harsh form. You might see that in a lab. So I trypsinized them, got them all up. What do I have to do after I trypsinize them? We'll go to this bench. Yes?
We have to add media to neutralize it.
Yeah, to neutralize it. So the serum deactivates the trypsin because the cells don't like being chewed up like that. So then what do? I pull all those cells up, I put them in a tube, then what did I do? Someone over here. Do you remember what you would do? I know you guys didn't do cell culture, but that's fine. We'll go back over here. Someone new, one of the ladies. What would I do after? Got a clue from your friend here. Spin them down, right? Yeah, so I have to centrifuge them down so all the cells pellet at the bottom. I pull out that media with the trypsin, and then I can resuspend them in fresh medium. And what I did, instead of putting them on a plate, I put them in the middle of, remember all those slides we coated with fibronectin? I dropped them in the middle of that. So right now they're sitting in the incubator and hopefully growing, kind of like sitting, well not growing, but sitting down and spreading out on those slides. So by the time we get to them, we'll be ready to stain them, I hope. So, great. So that's just the basics of cell culture, and that's what I did this morning. So now, like, you guys kind of did, I know you weren't able to image it, but you did like a live cell staining experiment if you were here on Thursday, right? You stained the cells, or Genevieve did, with a dye that fluoresces brighter green with calcium, and then you guys dropped water on it. You could see them get bright, and then they bubbled and exploded. So lots of times you want to do experiments where you do something to a cell, maybe treat it with a drug, or put it in a weird condition, and then you want to see what type of changes have happened in the cell. And that's where you would use one of the techniques we're using today, an immunoassay we call them. They're assays or like procedures where you're looking at how a cell has changed when you've done something to it. So I just want to do, this is like a really basic reminder, but there's some kind of key things about a cell you need to know for these. So we have our cell. What's in the center of the cell where all the DNA is? Just shout it out for me. Nucleus. Exactly. And then you have organelles in the cell, right? And so I'm just going to draw some little organelles in here. And then you have a cytoskeleton, so you have all these components that give the shape, and morphology it's called, that's like how the cell looks. So we have all these elements, so different things can mess up parts of the cell, so maybe you want to see if something happened to a lysosome or something happened to the Golgi apparatus with your treatment. But also cells, like, I just want to, like, very basic molecular biology. So in your cell nuclei, what do you have? What is that nucleic acid? What are the three letters that it is called? It's double-stranded DNA, right? So I'm going to draw my DNA here. So it's in your nucleus. And what is that DNA? Can someone just give me a really simple, not the technical name, although that's great, but like what is that? Like if you were telling like your mom or your grandma like what DNA is, what might you tell her? The genetic material, what's that mean? The building blocks of what make you human, yeah, the code I'm hearing. information. Yeah, so it's the directions basically for how to like make everything that a cell needs to function and perform all its duties. And do you guys know what happens? Like your DNA, it gets copied, right? And so when it gets copied, that's do you know what it becomes? What product you get? So the double strand splits. RNA, exactly. Fantastic. So we get a single-stranded RNA, right? RNA. And there's different types of RNA, but the one more interested in, it's called messenger RNA. That's the RNA that becomes protein. And does anyone know what this process is called when it goes from DNA to mRNA? I heard it a little bit louder. Say that again? Non-methylation. It begins with a T. Translate. And so I always remember, like, DNA is one language, RNA is another language. So what do you do when you're switching from like English to Spanish? To translate, right? That's how I remember which one that is. So you translate. No, I lied. I'm totally wrong. It's not. I'm totally wrong. It's transcription. Sorry. You transcript. So the first thing you do, so don't remember what I just said. Erase all of that. So when you go from DNA to RNA, you have to copy over what the code is. Sorry. These are a similar language. They're both genetic language. So we're transcribing. And then what happens to the mRNA? A ribosome attaches to it, and then what does that get translated into? It gets translated into another language. No, what? No, codons, but what do codons become? So they're like final product. Amino acids, and what do amino acids become? Proteins, exactly. Excellent, you guys. So we get to eventually proteins. And that's translating. So this is where we go from genetic language to an amino acid language. That's how you remember that. My coffee hadn't quite kicked in yet. So proteins are really important function of cells. They make the cell, they make all the parts of our body, and they do things. So different proteins activate different signaling pathways. So if the cell produces a certain type of protein, it can spit it out, and another cell can take that up, and it changes how that cell behaves. So when we're doing immunocytochemistry, what we're staining for, what we're looking for, are proteins. There's other assays where you might look at DNA or RNA changes, and it's the same principles that we're going to go through today, the same types of steps. But what we're targeting today are proteins. And so, yeah, are there any questions about any of that? That all makes sense? It's ringing a bell from biology class. And when you treat, like what we're usually interested in, you'll hear upregulated or downregulated a lot in the lab. So if you are doing something, treating your cells, and you get more of a protein, then you're upregulating that expression, and conversely, you can downregulate the expression. So when I toured you guys through the lab last week and showed some posters, and we were looking at that fancy microscope, something we were looking at in the cells was when we have our bone cells, when we flow on them, we stimulate them. So we want to know what proteins get upregulated and downregulated because of that stimulus. And then we were also treating our cells with a drug, and that drug changed how the cells, what proteins the cells produced when we stimulated them. And so what I've had students do for me, and I've done myself, is I would do those conditions to the cells. Like I would have cells I didn't flow on, cells I stimulated by flowing on them, cells that I treated with a drug, then flowed on, and then I would fix them. So like kind of freeze them in time, and then I would stain them to see how the protein changed. And one of the stains I used was LAMP 1, which marks lysosomes. Does anyone know what a lysosome is? Raise your hand if you know. Yeah, it breaks things apart. And this is super complicated. I won't go through all this pathway, but there's a type of, there's a pathway that's kind of like the sanitation system of the cell, where it collects stuff from the outside, and some stuff it sends back out of the cell, some stuff it recycles, some stuff it sends to like the trash, to the lysosomes to be like broken down. So I always think of it like the like dump trucks of the cell. But so some of those, sometimes there can be changes and you can get more LAMP1, and instead of becoming a full-on lysosome, those things can get, like they'll gather up packages and send them out of the cell. So I talked about exosomes, and I can talk to you afterwards if you want. But basically we were looking for LAMP1 expression, which marks a type of separatory lysosome, a lysosome that instead of going to the garbage, it's going to go out of the cell. So we were looking for LAMP1 to go up. And that was on the poster I showed. And I can show that again if anyone's interested later. But so that's what we're going to be staining for today. But our cells are they've just been on a dish. We haven't stimulated them. Okay, so that is just kind of an idea of why you might do an experiment like this. So then I just want to go over some of the basics of this protocol. So to detect our protein, we're going to use a thing called an antibody. And I'll talk a little more about those, I think, in a bit. But the main steps that we go through in all of these types of immunoassays, and we're going to talk about each of them as we go through. So I'm going to write like pre-step. And that's your sample prep. And this step's always really important because if you have a garbage sample, you're going to get garbage results. So how you treat your cells matters. If we were using cells that were covered in bacteria because they were all contaminated, that's going to affect our results. If our cells for some reason didn't stick well to the slide, that's going to affect our results. So you always want to make note in your notebook and be very conscious of the type of samples you're putting in and how you're treating them. And you think about, and you want to make sure before you start an experiment, you kind of understand what type of media, like all the things you guys have been going over, what type of media you need, what temperature your cells grow at, what the best cell is going to be for your experiment, etc. So if you have bad samples, you get bad results, which makes sense, but that's always an important one to remember. So then for all these, so then for the staining assays, we're not using live cells because we want everything to be at kind of the same time, so we want to freeze it in time. So the first major step in all these protocols is called fixation. And we're going to come back and talk about that in just a minute. With this step, sometimes you also have to do something called permeabilization. Let's see if I can. And then, so that's basically like freezing your samples in time. Then, all right, it's like I have a triangle that's accompanying me with those fingers. Then the next step in these is always a blocking step. And we're going to talk about that as well, what that means. And then you have a primary antibody step. I'm going to call it primary antibody incubation. And then you have a secondary. And actually, I know I'm starting to write low. If you guys can't see, you're welcome to kind of shuffle into this area. And then we'll go back to the benches. So secondary antibody, and this will be up all day. Incubation. And then the last step is, I'm going to write counter stain. Mounts. And then I'm going to write post, analyze, image and analyze. And this will be up all day. I know you guys are counting it down and I'm getting kind of low here. up until lunch, we're gonna get all the way through this part 4, and we're gonna do the last part in the second half of the day. So our sample prep, we went over, we like passaged our cells, I got them on slides. They're hopefully laying down nicely now. So what we're gonna do after we prep a couple of our reagents is a fixation step. And for fixation, what you're trying to do is preserve the morphology, which again is that kind of natural shape and, of the cell. And you also want to keep proteins from breaking down. Because as soon as you take the cells out of their nice media, their living condition, to try to start staining them, we talked about this, it's like they're on the moon, they're going to start breaking down, so they don't like that. And proteins can break down pretty fast. So there's 2 types of fixation. And so I'm going to go, I'll slide this back in a bit. Oh, I can actually go. So we're on fixation. There is cross-linking, so fixation. Type 1, cross-linking. And we're not going to do this type because you guys need to do a special training to work with. Has anyone ever heard of paraformaldehyde or formalin? It's what they put cadavers in too. But it has a very strong smell, and it's very toxic. So you have to do a special training to be able to work with that. But there's 2 things. One's called formalin, the common one for cells. I'm gonna just write PFA. That's paraformaldehyde. Aldehyde, I think is the last part. And it's a little less, I don't want to say less toxic. But what that does is it takes and links together parts of the cells and the proteins so that it's harder for them to break down. So that's a real advantage because the cell morphology, the way the cell looks, stays the same, and things stay pretty much in the same location. But when you do this, you can also hide where we need to get our antibody to be able to detect our protein. That can get blocked. So if I had, I'm gonna just put like a dot on my finger, like if this is where I need to bind to detect my antibody, if I cross-link, I cover it up so I can't get at it. So you usually have to do a permeabilization step, which we'll talk about in a minute, if you do this. Unless you're just staining something on the surface of the cell. The other very common type of fixation is using organic solvents. And the common ones are methanol, which I'm going to write MEOH, acetone, and ethanol, which I'm going to write ETOH. So these alcohols. And these can mess up the morphology a lot, but they're really good because they also permeabilize the cell, which we'll talk about in a minute, at the same time. So they're fast, and if you have lots of times a protein that's deep in the cell or in the nucleus, you use this type. But you always check, and we're going to look at our sheet, and then we're going to talk about why we're not doing what the sheet says. But so you fix your cells, and we're going to do this. We're going to use methanol, and you do it ice cold, which helps keep the proteins from breaking down. And then that also permeabilizes. So part of why we're doing methanol today is because it takes out a step. But the permeabilization, what that does is essentially puts little holes in the cell membrane so you can get your antibody and your stains into the cell. Have you guys learned how a cell membrane looks, or like how it forms? It's called a lipid bilayer. Have you guys heard that? Yeah. So you have a head and a tail, right? And I always say the tails are in. Is that right? Someone just say loud for me. Yeah. So what do we say the heads are? They are Are they? Hydrophilic, which means water loving, because they are by the cytoplasm. on the inside, which has water, right? Right, H2O. And whatever's outside the cell, which our bodies are full of water. So the heads love water, so they want to be by the water. So what does that mean the tails are?
Hydrophobic.
Hydrophobic, exactly. Which is water hating or fearing, exactly. So what the permeabilization does, and we do have something that's going to keep our cells permeabilized. It's usually like a detergent, like a soap, and it gets in between here and opens up these little channels so stuff can get in easier. So it's basically detergents, basically soap. They're usually very bubbly, like when you wash them, they look like soap. And that's actually why bacteria that don't have a cell wall, that's why washing your hands with soap and water is so important. That's what soap does to the bacteria. It gets into their cell wall, punches little holes, and then you kill the bacteria too. So always wash your hands with soap and water. So we're gonna do, this is gonna be combined in our methanol step. So after you fix your cells, the next thing you're going to do is it's called a blocking step. And what that is we're going to use antibodies to detect our protein. And I think we should actually talk about antibodies first. So in our cell, we have our, I'll draw a pie. So I'm going to redraw the cell here. So here's like our, what type of cell are we using? And do you remember the name? NC3T3, perfect. E1, yeah, perfect. And it looks, do you guys remember seeing it looks kind of like that, with little arms? And we have our nucleus in the middle, and then I'm going to draw, like, this is not to scale, but I'm going to draw a big lysosome here. So this is what we're going to be detecting with our LAMP1. And on our lysosome, we have some right here. So white and red is our lysosome. And in blue is our LAMP1, which is a protein that is on our lysosome. And the way we're going to detect this is, remember I said we fixed our cells, and then we put, because of how we're fixing it, permeabilizes the membrane, so I'm just going to put some little holes in our membrane. And then we're also going to use a thing called Teen20 in our PBS when we make it, which is a detergent which is going to keep that membrane open for us. And when we treat this with our primary antibody incubation, what those primary antibodies are is they detect this LAMP1. So they're going to go into the cell, and again, this is not to scale, I'm going to draw it really big. But basically, we have an antibody against LAMP1. So it's going to hook on to that LAMP1. And before we treat with all our antibodies, the thing is, the antibodies have things on them called an IG region, and it can stick to a lot of things inside the cell, but we only want to stick to the LAMP1. So the blocking step is we're going to cover our cells. Oops, let's go that way. Blocking. I'll write this in blue. Blocking. We're going to treat our cells. We're going to use horse serum. There's different ways you can block, but we're going to cover our cells with the serum first, and it's going to stick all over the cell. It's going to coat all inside that cell. So it's going to do two things. It's going to make it harder for the antibody to stick to things that it doesn't have a strong affinity, a strong like for. So the antibody will have to have a really strong connection to LAMP1 to knock off our blocking. So it's going to make sure that we get specific for specific binding. So it's going to cover a whole bunch of sites that an antibody might interact with, so that by the time we treat with our antibody will be able to outcompete for LAMP1, but it won't stick to stuff that it's not supposed to. And that helps, like all assays like this, what you're really trying to do is you want to increase your signal and reduce your noise. We know that if you guys have taken pictures, like that's what you do when you're messing with your iPhone or your Android and you're trying to make the contrast better so everything looks nice and sharp and not blurry. That's basically what the blocking is helping us do. Is it takes out stuff that might make our image look blurry or not specific. Does that kind of make sense to everyone? So that's what we're going to do with that step. And yeah. So I think that's what I want to cover up to here. And then we're going to make some stuff before we look at our cells. What time am I at? Just so I can have an idea of my cells. Or 10. Okay. So they've been like about an hour and a half. So that's good. Okay. So we're going to first make our PBS or TBS washes. And then we're going to get ourselves set up, and hopefully by then the cells will be ready to do the fixing and the blocking. So do you guys remember all the way back on May 12th when we made that 10x PBS? We're finally going to use it. And your parafilm was all really good because none of them spilled, which is great. We only had 6 PBSs, so I think this table and my table over here have TBS. 4 cells, like some labs use PBS, some use TBS. They usually kind of optimize. They're mostly interchangeable, so that's fine. So what we need to do for our wash is we need to go from 10x to 1x PBS. And actually, I'll go. We can head back to our stations, and I think everyone will be able to see me here. And we're gonna need gloves because we're gonna start making PBS. So I can't find the small and the large gloves. So if you need those are on the back wall. If you need medium, please take them from up here. There's at least one pen on every bench today. So we have some waste containers. I put tape on them. I want someone to label the big one liquid waste. You have a medium one. I want you to label that sharps waste. There should be tape on all of them. If there's not tape, there's tape sitting on your bench back towards the computer side. And you can label that, so it doesn't matter who labels it. There is one on each, there's a set of waste containers on each side. So the medium one is going to be sharps waste. And then there's a little one. You're going to write methanol waste on that. So someone, again, on each side of the bench, there should be a big container that's liquid waste. So all of our pour-offs today are going to be in the liquid waste. You have a medium one, it's a little bigger than the one I've got here. You're going to label that sharps waste. All of our pipettes, we're using the transfer pipettes, will go in that. And then you have a little baby one. We're just going to have a little bit, that's going to be methanol waste. Can someone tell me why we're separating the methanol waste? Raise your hand, maybe. Why can't we just put that with all of our PBS and stuff? Do you remember from 512? Yes. It has to be disposed of separately. Why? What is methanol? It is, what's the hazard? Does anyone remember? Flammable. Yeah. So methanol can't go down the sink. So when we use the methanol, we're going to pour it in our little container, and then we'll make a waste container. So where did Darnell poured this for me? Where did he pour it? Did he just do it out in the open when he made a bunch of methanol aliquots for us? He poured it under the chemical fume hood was where he made aliquots. So we're going to use it on the bench, but we're not really pouring it. Okay, so I just want to make this so we don't forget. What do I need to put on this chemical label? I need to put what? The hazard. So I have a checkbox on this one, so it's flammable, we said, right? Ignitable. What else do I need to put? There's 3 things that have to be on every chemical label. I don't need the date on the chemical label. Yes. The name. Can I write MEOH? No, because maybe the people disposing don't know that. So I have to write the common name, methanol. And then what else? The concentration, did you say? Okay, the concentration. So we're actually doing 100%. So they know like how strong that methanol is. So there's other things on this label I'll fill out later, but those are the 3 things like you always have. So we'll deal with that later. I'm just gonna put it in our hood. All right. Oh. Okay.
And we dispose of the rest of that.
Sure. So, all right, that's out of the way. So our PBS, we need to make 1x PBS from our 10x stock. There's a new jar for everyone. So you can just write directly on the jar 1x PBS on the clean jar that has the paraffin on top. Some of you have TBS, that's fine. If you have TBS, make TBS. So we have a 10x concentration. We want to make 1 liter of 1x concentration. So how much water and how much of my 10x stock am I going to need? You guys can write if you want on your notebook. Well, you can just work in your notebook. I'll give you all 2 minutes to think through this, and then I will take an answer. I'll give you 1 minute because you all look like you might know it. Huh? So we have 10x stock solution of PBS or TBS. We want 1 liter of a 1x solution of either PBS or TBS, whatever you have on your bench. So I want to know how much of the 10x solution we need and how much water we need. So take a minute or 2, look back in your notes. Okay, do we think we have it? Yeah.
100 milliliters of PBS and 900 of water.
Excellent. Is that what everyone got? 100 mils of 10x PBS or TBS, 900 mils of water. Is that, did anyone not get that, or want me to go over that math real quick? Does that make sense? Okay, great. So I just poured 900 mils. What is this called that I poured it into? Graduated cylinder. Great. And I'm going, so I did my water first, and then I'm going to carefully top it up to 1 liter, which is how many mils total? 1 liter. 1,000 exactly. So I top that up. So I'm looking for the bottom of the meniscus to be at the line. Perfect. So now I have 1x TBS. I'm going to take my, did I already lose my pen? Ah. So I'm going to label, what am I going to write on this? Where's my tape? So it's 1x, I have TBS. And then this I'll write my initials and the date on. So 1x TBS RLS 6918. And then I'm going to carefully pour this into my jar. And then I'll just carefully, PBS mixes pretty quick. So I'm not gonna bother to like stir it. I'm just gonna basically put my hand over it and shake it a little bit. So you all on each side of the bench, there is a fresh jar and a PBS. So you're gonna do the same thing. These 2 benches, there's a water right by the sink. These 2 benches, there's a water right here. So you guys, whether you're a pair or a trainer, you can kind of split up your duties. There's only 1 graduated cylinder, unfortunately, in the bench. I'll set this other one out. You're gonna get 900 mils of your water, add your PBS or TBS, label your jar, and everyone's gonna end up with 1x solution. Starting now.
You're going very slowly.
I know.
You could go a little bit more.
No, you didn't hit.
Yeah, I did. You have like
The bottom has
you have like
no.
990.
Not see that.
Oh my god. Pressure. Not like 999. Like bro.
Oh, you still need more. Your curve is like.
Oh my god. Is this Justin Bieber?
No, you're fine.
Okay.
Perfect. pour it. You want to pour it over the sink?
Kara, wait, let me hold the thing so you don't spill it.
By pouring it, does that mean you mix the solution? We don't have to worry about it?
All right, you guys have really good news. The cells look awesome. So I'm gonna take you in groups to go get your cells. Darnell, will you fill up the 4 cubes of ice? Just put one on each station. So I'm gonna just take you guys table by table. So we'll start everyone at this table. Have you guys, are you still making PBS?
Yeah.
Okay, no problem. Is everyone done at this table? Almost. Is everyone at this table? Okay, congratulations, you're first. I need you to take off one glove because we can't touch doors with our gloved hand, but we're going to be holding something else rather. So one glove off, and you're going to follow me, and I'll just circle through and we'll all get cells. The only difference between what you guys did on Thursday and what I did is instead of putting those cells in a dish, I put them on a slide that was coated. Alright, I'm just gonna have you guys file by one at a time to look at your cells and take a dish. Like I said, so you don't have, we're not gonna worry about whose slide is who. So go ahead and have a peek in. If you need to adjust, I put it on, so if you need to adjust the fine focus, the small knob, thanks so much, Sam. So you can grab that dish. And you see your cells are like right, so I just dropped them right in the middle. And you can head back to the lab. And then when you get back to the lab, I'll have the last person. Will you have Kyle send the next group over? Yeah, everybody next. All right, so let's. And you're allowed to move the dish around if you want, but you see your slides on there. That stuff floating around is just because I pulled them all up, so not all the slides have stuck. Can you see them? Great, so you can take that to your dish, head back to the lab. All right, so we've got our PBS, and a lot of you asked this. You saw some of the cells are kind of floating around, or they're like little balls, and because they haven't all laid down, so we want to get rid of that. So the first thing we're going to do is wash, and actually, Darnell, do you mind grabbing some paper towel and just kind of distributing some paper towel?
Yeah.
So we're going to wash, and then we're going to fix our cells. And so I'll just demo really quick. So make sure you can kind of see what I'm doing. If you need to step closer, you can. I know it's hard. So just remember, the way I set up these cells, they're all kind of in the middle. So that's what you're going to want to remember. So just really easy. I don't want to be sterile now, because we're going to fix this. So I'm just going to kind of tap this on the paper towel. You can also tap it in your dish. And then I'm going to pour PBS or TBS. I'm going to pour it on the side of my dish so that I'm not hitting the cells and knocking them off. And then just really simple, we're just going to kind of shake it, so we're just getting rid of the old cells. And then remember, I don't have cells in the middle, so I don't lose my slide. I'm going to put my finger on there and just dump that really good. So I dump out in my water waste, and then I'm going to dry it off. Be very careful that you don't accidentally flip your thing upside down and sting the wrong side. You want to keep it upright. Does that make sense? And then what we're going to do, so I'm going to actually take this out. This is the right side up. And I'm going to put it on the paper towel for a minute. And then we have ice distributed about. Is that right?
Yeah, we got.
Can you put one on each bench for me? But I need just one right now. And then actually, we need the methanol as well. So I need that out of the fridge. 2 on each bench, and I need 1.
In the ice?
No. Yeah, that's fine. Sorry. So I'm going to just put this, because I don't want them to dry out quite yet. So I'm just putting my cells in PBS. Will you pass me 1 right now and some ice? Thank you. So one ice bucket, and you can stick the 2 methanols, but I'm just gonna borrow 1 ice bucket.
This is a little one.
Perfect, I'll just borrow this, and then I'll put it on the bench back here. Okay, so we're gonna do the methanol on ice. So you guys will do it kind of all together. So I'm just gonna stick my methanol here. And on all of the benches are these transfer pipettes, and you're just gonna make it easier for us all to use. So I'm gonna grab a couple of transfer pipettes. The methanol's on ice. So this is the fixation step. And then we have a timer. And these timers, so there's 4 different channels. C is clear. You can set hours, minutes, or seconds, and then just a couple like cool things. So if I do 2 minutes and hit start, well, actually let me do that in seconds. I'll hit 5 seconds and hit start, and then it's gonna go off. When you're in a lab, you get used to like this noise. So if I hit stop once, it stops it. If I hit stop again, it resets it to the last time. That's really nice when you're doing like a whole bunch of washes that are 5 minutes. You don't have to keep setting it to 5 minutes each time. So that's just a little pro tip there. So, so I'm going to set this because I'm going to fix for I'll fix for 10 minutes. So what you guys will do is you'll all do the wash together, dryer thing, and then since you're using transfer pipettes, you'll all be able to get the methanol on about the same time and set the timer for 10 minutes. So again, oops, I'm going to dump this. I'm just touching the edge. I'm going to take this out. This is the top, but I don't want to forget that. So I'm just going to kind of tap it off, set it down, and then I'm just going to fill this with ice. So normally, well, not normally, you could use an ice bucket. You could do this in the ice, but we're just to make it easy for us. So I'm going to put my slide on the ice. I kind of want to push it down so it doesn't slide around. And then I'm going to take, I've already set my timer for 10 minutes. You squeeze the bulb and just pull it up. And then I'm just going to carefully, right in the middle of that, drop my methanol. And then I'm going to start my timer. And just keep an eye on it. If the methanol kind of comes off, like you should be able, you can just add a couple drops. But so that's what you're all going to do with your slides now. Dump it off, tap it on the towel, get the media off, wash it with your PBS or TBS, and then ice methanol, and we'll do the ice methanol all together. So you guys are on the same time. So here's your guys' ice bucket.
An ice bucket at each table.
Yeah, there's an ice bucket in the middle of each table. There's 2 things of methanol. The methanol is really cold. We've had it in the freezer for a bit. So you can take the methanol and set it in the racks, if that makes it easier for you both to access it. Yeah, and so there's 1 timer, I think, per table. So you can set like timer 1, timer 1, or if you're all at the same time. Oh, perfect. Thank you. Yeah. Excellent work. Yeah, and so we don't have to worry about being sterile, right? So we're gonna fix them. We just have to worry that we don't accidentally turn our slides upside down. Yeah, what you're doing is great. Kind of jam it in the ice so that everything's methanol. Yeah, is it labeled? Never have a tube of clear liquid that's not labeled in the lab. Yeah. We'll forgive you this time.
And we just leave to the side, right? And try some.
So you guys can, if you're ready, you can set like timer 1. But you don't need the top like.
We don't have to.
So do in groups. Like if you guys are ready, you're gonna do 10 minutes. Are you ready to fix? So set it up for 10 minutes. Yeah. And so they'll be on timer 1 and that goes off, and we'll be on timer 2. Like you guys all do it together. I'll show you how you can tell which timer's going off.
Just set it and then we'll start.
Yep.
Face up, right? I think I put too much on it. Just clear it. Let's see.
What was that? Oh, they can do it in groups. Yeah. You don't have to put the top on. Yeah. So we don't have to be sterile because they're fixed. The methanol will kill like any bacteria. There wasn't bacteria anyway. But yeah, so you don't have to be sterile. You want to be tidy when you guys work at the bench, but this does not have to be sterile.
Just 30 seconds. See, it's just happening like that. Do it.
Just put
Yeah. Okay, so I can start? Yeah.
You guys start time 2 for you guys.
I think we started, what did you start? We started
Well, you have to drop it.
Yeah, if it's
It doesn't matter.
about 10 minutes, then we're not publishing these results. So everybody, actually, while we're standing here, will you please label your methanol as well? Full word, not MEOH, methanol, 100%, someone's initials and the date. So everybody's in methanol, right? So we'll gather back over here. My timer's about to go off. Great. And we only have 2 of these. So this is good for our own groups. So timer goes off. When you have multiple timers set, so let's say I have timer 4 going and timer 3 going. So the number that's flashing, you are not on that timer currently. The number that's not flashing, you see that T3? That's the one. So when you have a timer going off and you're like, which one's going off? If I click to T4, that's stopped. And when I just hit stop, it stops the timer. So now T3 is flashing, so that's not the timer on. So sometimes you have a lot of experiments going. You might have 4 timers going. You won't initially, but when you get really good, you will. So that's how you know like which timer is which. You can always write it down too. Genevieve and I both do this. We make notes on our lab bench all the time. So my methanol's done. So I'm going to carefully dump this and kind of dry it a little bit at the end there. But dump any of it that's there into my, what's it called? My methanol waste there, and then kind of dry it off. This ice, I'm just going to dump in the liquid waste because it's there and it's easy. And then we have Kim wipes on the table. So yeah, I'm going to borrow your guys'. You can just take a Kim wipe, and we're just going to use this as our staining dish. I'm just going to kind of dry that off a little bit. I'm going to put that there. Actually, before I do that, so we're going to use, it says step 5, using a PEF pen, set up regions for staining. So these are really handy. You won't always have these. You can just drop things in the middle, but when you're doing long staining or you want to set up regions, these are super handy. It's a hydrophobic pen. So what does hydrophobic mean again? Yeah, water varying. So it's going to repel water. So what you can do, I'm just going to kind of, it has protein that's hydrophobic. So I'm going to take my Kim wipe. I'm just wiping off the back so you can kind of see what's going on the sides. And I want to dry my slide just a little bit. And then I'm going to draw basically like a border right in the middle where my cells are. You don't want to push too hard, but do you see how I have like a square now? So I'm going to concentrate all my reagent. And what it's going to do is it's going to keep my reagent, all the stuff I'm putting on, from spilling out. I only have 2 of these, but it's good because we're all slightly different timing. So you guys can come up and draw them. So I'm going to do that. I draw that on, and then it says that I need to wash in PBS 3 times. So I'm going to just pour this times 3. It says 1x PBS times 3. We don't want all the extra stuff sticking around because that's going to make more background. So we want to make sure we've washed off really good. So what you'll do is we'll do 2 rinses like that, and then you'll just leave your stuff in in PBS till everyone's in the same spot before we go on to the blocking step. And then we'll do the blocking step all as one. But so, yes, so I'm going to actually do that now too. And we'll do the blocking all as one. So I'm just going to leave it sitting in PBS. So you know, when you're going through protocols, it's always nice to know where you can stop if an emergency happens. Because there's some steps where you can stop on stuff, and there's some stuff where the timing is really essential, and it will change what your data looks like if you leave it longer or shorter, and you're trying to make comparisons. So in general, any wash step, you're okay if you go over. So if you have to go to the bathroom, if there's a fire drill, if there's an emergency, you have to leave the lab, if your PI comes in and you suddenly have to have a meeting, try to get to the next wash step, and then you can usually leave it there. And what I'll do in my notebook is I'll just make a note like in wash for 20 extra minutes or whatever, so I know. But that's usually fine. Things like primary antibody, secondary antibody, stain, like the important things, those are usually timed, and especially if you have 50 samples you need to stain, maybe you can't do them all at once or do them over multiple days. So the staining steps, you want to be as close to exact for each of those. Blocking, usually you're okay, a little extra time as well. But yeah, so I think our timer should start going off soon. So you'll go back, so you're gonna, you can double check and make sure the methanol hasn't slid off. If it has, you can cover your cells again with methanol. And then if your timer goes off, carefully take the methanol and put it in that methanol waste cup. When you get to the washes, you can do 2 quick rinses with PBS, and then your final rinse, just leave it in PBS until we're all done. After the methanol, you do draw the path pin. So if you're ready to draw a path pin, you can come to my station with your slide, and I have 2 pins. I can help you. So yeah, you guys, it's super tired. So yeah, try out the finish, wipe the back of your lecture, do the edge and strip it off. Yeah, sorry, we did it. I'll put it half. Yeah, that's great. So someone over here was done next.
They do the PBS and then I actually drew on it, but it's not a protocol that's out there.
Yes. Great. Just pour it in your dish is full, and then swirl it around and dump it off. Yeah. But you notice how it stays just in that square because the hydrophobic barrier is making it more. You guys have a pen over here, and then you guys, just pass it down to your table. You don't want to knock them off, right? And then you both unpack them. When you're done, pass it behind you. That's it, boy. It's right there. Yeah, and some of you are noticing, so your liquid, right, it repels off of that pathogen barrier. So you'll definitely have to switch around. You want your cells to get wet, not the outside, right? So get more PBS on there. Yeah. You want you're washing your cells. But you want to fill that not on the door on the side, and then squish it over. Yeah, because you don't want to knock them off. So you something like that. Yeah. And then you guys do you have to go back? Don't let it dry. Yeah, so now you don't want your slides. Once we're to the step, we don't want our slides to ever dry. So if you're just waiting for something, once you get that packing on, keep your slide in PBS. Because if those cells start to dry, they're going to take out the stain weird. They're going to take it out weird. And when you guys are done, just do the pen. And you don't have to be conservative with the PBS. Obviously, we have a ton more. So if we need to make more for washes in a bit, we can. So don't be stingy with your wash.
You can see it. You can see it. No, you're fine. Look, there's a little square. You can see it. When you put it in the PBS, you'll see it. Actually, if you want to go again.
Oh yeah, thank you.
Should I tap it right in here?
Oh, we have both of them next.
No, actually.
Oh, yeah. Could you imagine me with more caffeine? No! I always show myself about 1.8 ounce glass in the morning.
We're going to pray
Too high for otherwise. Okay, so everyone has their patch pen? Yeah? Okay, great. So, we fixed our cells now, so now they're like essentially dead. They're like frozen in time. They're not going to grow anymore. And we have them, we wash, put that path barrier to conserve our reagent and just make sure we're working in an area. So what I would do lots of times when I'd have big slides with a lot of cells that I would flow on, and I would use that path pen. I'd make like several different regions, and then I would have, we call it a negative staining control. So I'll actually, we're not going to bother with that today, but when you're doing a real experiment, you always want to think about how you're going to control it. So you know if you see a signal, if it's real or not. So I'm going to put up a note about controls here. All right. So experimental controls. Oh, actually, you know what? I'm going to do this in just a minute because we have a 20-minute step, so we can talk about controls then. So that was just a teaser. Okay, so everybody's got their stuff in PBS. We've done a couple washes. So I've got your guys'. Everyone else, I put one of these out. It's normal horse serum. So it's sitting by the microscope. So each bench has one. This is part of our secondary kit. So on this page, I put 2 data sheets. This one that's up and down. This is our secondary kit, and we're looking at the 2.5% normal horse serum. This company is really nice because we don't have to mix it or anything. You can also make your own blocking solution. This is just dropper bottles. So we can just squeeze a drop right into our path pin area. The serum, we'll get into this a little bit more in a minute, but the serum, if you're using serum, it's what your secondary antibody is raised in. So if you have your pen out, well, we'll talk about that. You can make that as a note. But so your secondary antibodies are raised in horse. I'll talk about what that means. There's other non-serum blocks you can use. So you might have a protocol in a lab you go into, and they use BSA or milk, something else. So that's fine. But the idea is that we're blocking non-specific binding. So you don't need to move from your section. I can just, I'll pull this out. But what I'm going to do is I'm going to dump this into my waste. I still have my finger on my slide, and so just kind of tap it on my paper towel, tap my dish, so that it's pretty dry. And then I'm going to take this dropper, and I'll just kind of hold this out here so you can see. So I'm just putting like 1, 2, 3, we'll say 4 drops right in the middle of my path barrier. And to spread that out a little bit, I'm going to take a fresh transfer pipette. And I don't want to scrape myself, so I'm just going to kind of carefully push it to the edges of my barrier so that that whole region that I circled out is covered. So I'm careful just to go over the top of the liquid. Do you guys see that? So I'm just kind of filling up. And what I end up with is my square that I made is full of that horse serum, that blocking. So now this is the only area of my slide that really matters. And so then I'm going to cover this. The only reason I'm covering it is because, like, I don't know, if some dust falls down or something, I don't want to mess up my sample. So I'm just going to cover it. I preset my timer for 20 minutes, and I'm going to start that for 20 minutes. So you guys are going to do that step, and then we will chat a little bit more once you guys get, this should be pretty quick. So we'll just kind of talk through it. So everybody can dump, tap your dish, and then you'll drop that horse serum. 4 drops and pass it to your friend, and then you can take the transfer pipe, fresh transfer pipette, and kind of carefully try to spread it out.
When you're spreading it out, make sure you're not scraping the slide. You don't want to like actually scrape off the sides.
And so then, so everybody has serum on their thing? Awesome. So then let's set some timers. Actually, I'll just set 1 timer for everyone, that's fine. Timer 2's you guys. So we're all in serum about the same time. 20 minutes, if we end up a little over, it's okay. So I got a timer going for everyone right now. We'll collect up those horse serums. So this, I brought out of the fridge, and then we're gonna put it right back in the fridge when we're done. And so then the next thing we're gonna do, we're gonna prepare our primary antibodies. So I think this makes the most sense. So don't move your dish around, just let it sit there. You don't want this stuff to come off. So we're gonna prepare our primary antibodies, because we've got about 20 minutes. I think it will take a little bit of time to do that. And then we'll go into, like, our stuff's gonna sit there for a long time in primary, and we can talk about what the heck that means, primary antibodies, and all that up to lunch. So these were frozen in aliquots. So a lot of times you get your antibody and you get a big stock of it, but you don't want to keep freezing and thawing things because that can break them down. So I took, this morning I thawed them. It's 25 microliters in each of these purple things. I want you to be careful, but I'm going to set, so go ahead and flip. Oh yeah, there's this too. All right. So you're gonna flip your block so that the little circles are up, and I'm gonna give you each a primary antibody. And then I need everybody to have 1 orange juice. All right, so next I want you to pull out your LAMP1 antibody data sheet. And in between, we'll talk about this after we've made it, but in between all the microscopes, we have a more detailed data sheet that we're going to talk about. But I just want the one I printed for you. And so this is the name of the antibody. We bought it from a company called Abcam. They're a popular antibody company. They make good antibodies. It tells you the product name, that it's a lysosome marker. The description, it says a rabbit polyclonal to LAMP1. So that means the host species is a rabbit. We're going to talk about that. Your host species, you should try to avoid it being the species that your cells came from. So our cells are mouse cells. So we don't want our host species for our antibody to be mouse. It can work, but it's harder. And we don't want our secondary and our primary to be in the same host species. I will go over all of this more, but ours is rabbits, just so you know what you're kind of looking at on these data sheets. It tells us the species reactivity. It reacts with mouse, cat, chicken, hamster, cat, dog, human. It has a whole list of things. So those are all of cells from all of those animals our antibody will work with. Some antibodies will only work on cells that have come out of a mouse or a human. So you have to always check that. Just because you have an antibody for LAMP1, you have to make sure it works with the cells you have. Then it gives us what we can use as a positive control. We'll talk about that in a minute. its form is liquid. Sometimes they come as a powder, and you have to do something called reconstitute them. So you have to put them back in liquid. It tells you how to store it. Down here it says to store at -20 or -80 and avoid freeze-thaw cycles. So that's why I made it into those littler bits. So then I only have to take out how much I need. I don't have to take the whole thing out and thaw it and put it back and thaw it and put it back. And lots of times when you're a young person in a lab, you end up aliquoting a lot of stuff for grad students.
Oh, yeah.
And that's just pipetting over and over, but it's important. The clonality is a polyclonal. We'll talk about that. The isotype is IgG. And then it has a list of applications. At the top it says tested applications. So you see these abbreviations a lot. These are all different types of aminoacids. So IP, I forget what IP is actually. Immunoprecipitation, yes. IHC is immunohistochemistry. That's staining tissue that's on a slide, not cells. When I did the tour of the lab and showed some pictures up on the microscope, that was tissue. ICC, that's what we're doing, immunocytochemistry. They have an ICC/IF. IF is immunofluorescence. That is exactly what you do. WB is Western blot, and IHC-FR is immunohistochemistry on frozen sections. So you'll get used to seeing these, but no, like some antibodies will only work for a Western blot. Some antibodies only work for ICC. So you always want to look up the data sheet. So if you get an antibody, you can find these just by looking at AB 24170. You always want to look it up so you just know what you're working with and if it's going to actually work for what you're supposed to be doing. And then if you turn to the back, they'll often give you, Abcam's great, they'll give you a list of what you can use it in and some suggested concentrations to work within. But you always have to figure out when you have a new antibody how much of it you need to put on. So if you put too much, you're going to get an oversaturated signal and you're just going to stain everything. If you put too little, you're going to get an undersaturated signal. You won't be able to see what you want. So sometimes they say use an assay-dependent concentration. That means somebody in your lab, maybe you, has to do some work and some experiments to test a whole bunch of different concentrations and see what's going to work. This antibody's old, so I'm hoping it's going to work. When we did this before, I looked up in my old student's notebook, we figured out that a concentration of 1 microgram per mil worked. I've diluted this antibody. So that means we're going to do a 1 to 200 dilution further because this is currently at 200 micrograms per mil. So you can make a note of that on your protocol or on the data sheet or in your notebook. These are things you write in your notebook. So our stop concentration is 200 micrograms per mil. And we're going to work we're going to apply a 1 microgram per mil concentration. To our samples. That means we need to dilute this 1 to 200. So there's enough of this here for everyone to make 1 mil of their primary antibody, which will be more than enough to cover our sample. Remember I said you can never escape this equation when you're doing biology? So our initial concentration is 200 micrograms per mil. We don't know what volume we're putting in. Our final concentration is 1 microgram per mil. And we're going to do that in 1 mil, which is, 1 mil, that's fine. So 1 divided by 200, that's horrible writing. Sorry guys. Okay, so that equals X equals 0.005 mils. How many microliters is that? Which way? We're going from something big to something smaller. Which way do I need to move my decimal point? The left, really? Right, there's going to be more of the smaller thing, right? We're going from something big to something smaller. So that's going to be, and how many, this is 10 to the minus 3 mils? What's micro? So how many decimal places am I moving? Or sorry, this is grams, yes. Minus 6, okay. So I'm going, so X also will equal then 5 microliters, right? So 5 microliters, my total volume is how many microliters? 1 mil is how many microliters? 1,000. So how much of my diluent am I putting in? Like I'm gonna mix it, I'm not mixing it into water this time, but how much? So I have to subtract 5, right? So final volume of 1,000, so that means I'm gonna put 5 microliters of this into 995 microliters of diluent, I'm gonna call it diluent. Sorry guys. I could practice my word writing. Okay. That's more or less how it's spelled. Does that make sense to everybody? Yeah? So this is so I said it's a 1 to 200 dilution. So for every 200 microliters, I'm putting 1 microliter in of my stock. So a lot of times you'll see that written on a protocol, 1 to 500, 1 to 100. So that's where I got that 1 to 200. So the diluent we're going to use is our PBS, but we're going to add a detergent to it to help permeabilize our cells. So we're going to add between 20 to our PBS or TBS. So let's distribute, actually, do we have, will you grab the 50 mils is fine.
Okay.
Yeah. Will you please distribute a 50 mil tube to everybody? And all done on this. So we're gonna make our diluent, which is TBS or PBS. So I'm gonna write TBS-T, and it's going to be 0.2% between 20 in TBS. So that we're gonna make 50 mils. So 50 times 0.2%, I need to times by 0.002, right? So that means I'm going to add 0.1 mils, which is 100 microliters. Have I done this right? Yep. No, sorry. Let me just do this real quick. Yeah, okay. Okay, so I'll write this out too. Sorry. We're gonna make for PBS or TBS-T, I'm gonna write PBS-T. So this is our 1x PBS with our tween 20, which is what that tube is that you guys pulled out with that weird yellowish stuff in the bottom. Tween 20 is a detergent. It helps put these holes and maintain these holes in our membrane so our antibody can get in. So we want it's tween 20 equals 0.2%. So we're gonna make 50 mils of this. So I got 50 mils times, it's percent, so I need to move the decimal 2 places. So 0.002 equals 0.1. So that's 1 mil, 0.1 mil, which is how many microliters? 100 microliters. Of between 20. Okay, so because 100 microliters is very small compared to 50 mils, we're not gonna worry about doing like 49.99 mils of this. So what you're gonna do, you have a small cylinder, so you're going to do this in pairs. You've got your 1X solution, you're gonna pour 100 mils of it into your graduated cylinder. Then you'll, with your friend, one of you will take 50, the other one will take 50. Or maybe your lab enemy, I don't know, but you're gonna share. And if you overshoot it a little bit, you can transfer back and forth. All right, so I got my 50 mils, and then I'm gonna add my tween 20 to this. So you each have, it looks like this. We aliquoted it earlier for you. Notice how it's called viscous this is. Viscous stuff is hard to pipette. So I'm gonna show you a trick. You all have a 1000 here. So I'm gonna take my pipette down to 100. A trick when you're pipetting viscous stuff is to open up the tip a little bit so you've got a thicker area you're working with. So we'll pass out some scissors. So I'm just gonna actually cut the end off so this is thicker. That's gonna make it easier to pull up. And this is a mess to work with, so keep it over your pad. And then I'm gonna pre-open my tube. So like I have these ready to go. So I push down to the stopping point. I'm gonna pull up to 100. It's not very much in this tip. And immediately transfer it over so it starts to drip. And then I'm gonna pipette up and down several times. Dispense that all. And then tip goes in the sharps. And then, so that's my block. It's okay if it's just a block a little longer. So I'm actually gonna start a timer. If you start from 0 and hit start, it's gonna count up. And I'll just make a note in my notebook how long I blocked for. So then I'm just gonna shake that like crazy. And you see it gets kind of soapy because it's detergent. So make sure it's closed tight. And now we've got our diluent. So that's what you're all gonna do. So you're gonna fill up to 100. You're each gonna take 50 of it into your tube, and then you're gonna add 100 mils of your tween 20. Does that make sense? All right, so everyone let's make some diluent. I'll have out some scissors. Yeah. There should be 2 graduates. There's only 1. Okay, here. So here's another one that's got like about 50 in it. You can take a little out, but you guys can start with that one. So that one is TBS, just so you know. Or yeah, so let me get this one there. Sorry, no, I'll take that. Yeah. So this is TBS. You're my last friend. That's TBS, and then I'll take it over.
Yeah.
This is
I think she gave you some. Yeah, they're right there. You guys are
100, yep. So it's 0.2%. You guys are so impressed. You remember being in here the first week?
I still have support.
Trying to pipette and it was so confusing and it took us like 2 hours to make PBS. You guys are doing awesome. What's your turn? Oh, you want, okay. Yeah. Yeah, we'll get to that when we get the secondary antibodies. Yeah. We gotta hold it.
Oh, yeah.
Yeah.
It's so full.
Yep. couple more. Okay, so just to like reiterate, so it's clear, because I know I did that maybe math in a backward order. So we need to make our primary antibody that we're going to put on our cells and let them incubate in primary antibody. To do that, we're diluting a stock concentration of antibody that's at 200 microliters per mil, and I have previously determined by doing optimization experiments that the staining works best on these cells at 1 microgram per mil. So that was the CV1 equals CV2 math we did. But we need to put that antibody that we're diluting into something specific. We're not putting it into water. We're putting it into our buffer, which is PBS, 1x PBS, with tween 20 added to it. And what that tween 20 is doing is it's keeping the cell permeabilized. Because it's got detergent in it, so that detergent's getting into the cell membrane and making sure that there's holes so that our antibody can get into our cell. And that was the math on the right-hand side. We were just figuring out how much tween 20 we needed to put into this. So now that, so this is called a diluent. It's what we're putting our antibody into. Later we'll put our secondary antibody into this as well. So now this is the 995 microliters that we need. So we're going to take our diluent. I need the, oh no, of that. So we're going to take 995 microliters of our diluent. We don't need to cut the tip anymore because we diluted that tween 20 down. So this isn't viscous, right? This is more like water. So that's easy to pipette. Tween 20 and Triton X are the common things you'll find in the biology lab. Those are hard to pipette, and that little trick of cutting your tip helps a lot. So I've got this at 995. I'm going to take my 1.5 mil Eppendorf tube that I pre-labeled. Actually, this is 2 mil tube. I'm going to take, so I push to the first stop. I put my tip in. I slowly draw up. Oops, that was air. Sorry, I put it in my diluent, which I actually want. So I've got my 995. I make sure there's not any bubbles in there. Then I dispense it all the way to the second drop. I adjust my tip. And then how much am I adding? So I'm going to put this lid on. How much of my primary am I adding again? 5, yeah. So we have these, I think some of you have 1 to 10s and some of you have 2 to 20s. And I think I put the right tips that work. We have less of these, so we'll have to share. But this is a 1 to 10, so I'm going to put this at 5 microliters. So I'm pipetting up and down several times so that I mix that antibody before I draw it up. And then I draw it, but last time I only pushed to the first stop each time I did that. I'm going to set down my little aliquot. And then I want to watch this go in. This is a small amount. So I put it in my liquid. And again, I pipette to the first stop several times to mix it. And then I dispense all the way to the bottom, pull out, and eject my tip. So that's my 1 to 200 dilution, or 1 microgram per mil concentration. I have, normally in the lab, I would like just vortex it. So you guys can come up and use my vortex as you get it. I only have 1. So, and then that's ready to go. So then I'm just gonna, I'm gonna close my secondary, and I'm just gonna set this in my rack until I'm ready. All right, so you guys are all gonna do that. We'll kind of pass around pipettes so we can get through it faster. Yeah, and then once you've made your primary, you can come up and vortex it, so it's just well mixed, and then back to your bench. So I know what's going on. Right. Yeah, no, if you feel like, but you'll see, when she pulled that slide, you'll see it looks like looking for that. And then you'll be more confident, you know, that'll be super fine. Any questions? Don't wash after is blocking. Because you want the blocking still there. You want your antibody actually to have to compete with that block a little bit. Because you only want the antibody to go where there's strong binding. So this antibody should only go where there's lamp. And it likes lamp and it binds strongly to lamp. So it will knock out our block where it's there. But whereas a weak interaction, our block will win and our block will stay. So we're not going to wash. So you guys can actually all stand at your bench. We'll just do this together. It's not so hard. You can make a note for yourself. So I just started the timer counting up at 20 minutes. So I knew how long extra our stuff was in blocks. So you can make a note in your notebook as you're writing today's protocol. So we ended up blocking this for 16 minutes extra. So a total of 36 minutes was how long we blocked. Which is, it's fine. You can, sometimes you block things for longer. So is everybody made their block? Their primary. We've got one more person. Okay. All right. So I always double check. So you want to make sure your slide that you still have liquid covering it. Because if the liquids come off, your stuff might have dried out and it might mess up the staining. If your section dried out, you would make a note in your notebook, think it dried out and blocked, so if your results don't look right later, you'll know. So we can do this all together. We're gonna take our slide out. You're just gonna tip it onto a paper towel and tap it, so you tip off the blocking reagent. So you see I still have a little bit of block on my slide. It's not completely dry in that square, that's okay. You just want to tip off the excess and then put it back in your dish. Then you take your primary, you take a fresh transfer pipette. So everyone grab a transfer pipette out. And then you're going to carefully, you can pipe it up and down a couple times to mix it again, but you all just vortexed it. You're going to draw up a little bit, or it can be all of it, doesn't matter. You're going to draw up your primary that you made. And you're going to drop it right into that pap pin square. And you can fill up that square, but you don't want your liquid to go over. You'll see if it spills over, it should pull away, but just make sure you have a nice kind of like rounded dome of liquid in that square with your primary. And then I'm going to just toss out my transfer pipette, and I'll keep my primary nearby in case it gets dry, but it should be fine. So we'll walk around and see you guys do that. And then I'm just going to start a timer counting up. So that I clear my timer completely and then hit start to count up. Yeah. It's okay. It's okay. It's not horrible if there's some bubbles. Like, try not to have bubbles, but it's okay if there are. You put it in your mouth? Don't put anything in the lab in your mouth. What? Okay, be careful. So that. Great. So the way to help not get bubbles, a little trick I should have said, you don't want to overdraw. You basically don't want air in your tip. You want liquid all the way to the bottom. So when I draw stuff up, like if I accidentally draw up to there, see how I've gotten bubbles because now I've got air? Like I'll push this out until I get to where the liquid's like right at the tip, and then I'll put the liquid on. So when you draw things up, you want to keep pressure on this so you have liquid down at the bottom. As soon as I release, then I fill air up in the tube, and that's how you get a bunch of bubbles. So they'll pop. It's going to sit in primary for approximately an hour and a half to 2. It's fine. So is their sections covered? you're within the barrier, great. So you just put your lid on it, just in case some dust falls or something happens. And then, what's my time? So everyone has their primary on? So in this step, what's happening now is we have our primary antibody against LAMP1. We have our, it's in something that has a little more fertilizer in it to keep these holes in the cell. So that antibody over the next couple hours should find all of our LAMP1 sites and stick to it. And that's how we're going to detect our LAMP1. So how do we get these antibodies, or what the heck is this antibody that I keep talking about and teasing? So anybody, does anyone kind of want to tell me just like how you would tell your grandpa this time about antibodies? I don't know why, old people are very smart as well, you guys. Just, they probably know. Do you have, have you heard, what the heck's an antibody? Do we make them ourselves? Yeah, what are they against, usually? Infection, a disease. Yeah, they're against things that, are foreign to our body, right? So they're a way the body's immune system can detect foreign stuff and mark it so we can get rid of it. Antibodies also train our immune system to be able to detect in the future if that foreign stuff comes into us, so that the immune system can get rid of it. So the way we get commercial antibodies, like the one we're using, LAMP1, so I said it was a rabbit polyclonal. So the way they get that is they inject the LAMP1 protein. They purify out just, they go through it says to purify out basically just these blue dots from my cell. And let's see if I am not an artist. You inject it into an animal, in our case it's a rabbit. And the rabbit's immune system sees this foreign protein and starts making antibodies against it. And then we can draw out blood from the rabbit and separate out those antibodies. And those antibodies, they're specific now to LAMP1. They'll be able to capture, they've been modified to capture and identify that LAMP1 protein. And a polyclonal means there's a lot of different kind of versions of that antibody that come out. So this LAMP1 protein, it has several different amino acid sites. So there'll be one antibody that might detect the end, part on the, well, you don't need to know all. But there'll be another antibody that's shaped like this. The antibodies will be able to bind to several different places on our protein. So I'm going to draw our LAMP1 protein as kind of a set of amino acids. Kind of like that, I guess. And then there'll be a site, this antibody will match up with the site here, but there'll be another antibody the rabbit's immune system would have made that might match up over here. So that's a polyclonal. Poly means many. A monoclonal antibody will only recognize one specific site. So monoclonal antibodies are very specific, but your signal's often not as strong. They're less sensitive. But polyclonals are more sensitive because they'll recognize a lot of different sites. And it depends what you want. Yes?
Can I ask two questions?
Yes.
Which cell type, spoken of ad nauseum, makes the antibodies? Which cell type that I've mentioned several times when I've given these presentations makes these antibodies? Which type of white blood cell? 100 extra credit points. No, wrong letter. B cells.
B cells, yeah.
Second question.
Yeah.
What preventative public health measure is this whole process reminiscent of? It's seasonal. I think seasonal vaccines.
Yeah. Yeah, vaccines. So vaccines, you inject like a protein or something from the virus or the pathogen into the body. It's deactivated, but the body recognizes that and makes a bunch of antibodies. Yeah, exactly. And so you can also, so it's much more expensive to do this, so you don't get them commercially. You can also make antibodies, you can engineer them and make cells make them. So my company does that. My company makes antibodies. So in the stuff I study, I actually use, it's more complicated than we have to go into, but I basically have the DNA instructions on how to make antibodies, and I put those into a cell, make the cell make the antibody for me, and bypass the animal. The commercial antibodies, you have the animal. So the rabbit makes our antibody. There's goat is a common one, sometimes mouse, but it's usually rabbit or goat. Then we draw out and we purify these antibodies, and that's what we have as our primary. What's my time?
Right now, it's
Okay.
5 to 1.
Okay, so I'm gonna just tease the next part, and then we'll go to lunch. Just so now this is our so that's how we got our primary antibody that we're attaching to the lamp one. Are there any questions, or does that kind of conceptually make sense? Great. So our primary antibody, it would be really expensive if we, like, my company will modify a primary antibody and your steps done there. But that's, it's very expensive to do that. So now our next step, the secondary antibody incubation, when we come back from lunch, is now we're gonna detect our primary antibody. So what that means is I can have an antibody, so this rabbit, because this rabbit made this antibody, the rabbit wants to keep the antibodies in its system, because if it gets injected with LAMP1 protein again, it wants to get rid of it because it's foreign to it. So on this antibody, there's an IgG immunoglobulin region that's unique to rabbit. And that so like we have human IgG, there's rabbit IgG, there's mouse, like whatever the animal is. But the IgG on the antibodies that come from the rabbit it's telling the rabbit's immune system, this belongs to me. You can keep this antibody in here. Get rid of the foreign protein, but keep anything that's marked with IgG. Those antibodies are okay. So the secondary, what our secondary is going to detect actually, let me make that IgG. I'm going to make it just like a green thing. So here's our IgG. The secondary we're going to detect, it's going to be against the rabbit IgG. So that means financially that I can make antibodies in a rabbit against LAMP, against sclerostin, against rank log, against a whole bunch of things, and get all those antibodies and purify them. But I don't have to make a way to detect every single one, because that'd be super expensive. I can just detect anything that came out of the rabbit. And if I know I started with a LAMP1 primary, my secondary is going to detect that it came out of a rabbit. So that's why I said the serum, the primary and secondary antibodies have to be from different things. So our secondary antibody is raised in horse. Sorry, that's a little brutal. And it's against, so the horse gets injected with rabbit protein, rabbit IgG, and then it's like, whoa, this is foreign. Rabbit IgG doesn't belong in here, and it makes antibodies with its B cells, make antibodies against the rabbit IgG, and then we take those out and purify those, and so now we have a secondary that's anti-rabbit. So it's going to detect this one, and this. So this is the blue is our anti-LAMP. I'm going to write a little alpha there, anti-LAMP. And our secondary is, I'm going to erase this map because I think you guys got this all and I want my picture. If you need me to do that math for you later, I will. This is our secondaries anti, I'm going to write R for rabbit, R-I-G-G. and it's gonna be the secondary that we actually detect. So now we can make like a broad spectrum secondary that will detect a lot of different antibodies. And our particular secondary has a fluorophore on it, and we're gonna talk about this in the afternoon. But it has a fluorophore on it that's gonna, when we shine light of a particular color, it's gonna fluoresce red. So this is, it's called 594, I think. What's it say on this sheet? 594, yeah. So this is 594, so that's the color. I'm gonna write red. And that's how we're gonna detect this later. So it's an indirect detection method. So we're detecting our protein first. We find it with the primary antibody. That's what we're doing right now. In the afternoon, we're gonna detect our primary antibody, which is attached to our protein, with our secondary antibody. And that secondary antibody is what we're gonna activate with our laser light. Sorry, it's not a laser here, but with our fluorescent light, and visualize in the afternoon. I saw a hand, yes?
How do you extract antibodies?
So they draw out blood, and then it's in your serum. So like, has anyone ever given blood or had to have blood taken? Yeah, some of us? Yeah, so when you draw out your blood, it's like kind of red and liquidy. They'll clot out the red cells, the red blood cells, like clot on the bottom, you spin out those, and then you get this clear serum on top, and that's everything else that's in your blood, including all the antibodies that your body's made. So there'll be tons. And then you have ways that you detect, like you pull out, so there's probably in an assay where you look for the rabbit IgG and pull out all of the rabbit antibodies. There's ways to purify from there. Yeah, yeah. Any other questions? Good. I know this is a lot, but is it sort of conceptually making sense? Excellent. Great. All right, you guys ready for some pizza? Perfect. So I started our timer just counting up. The primary, we're going to do between 1 and 2 hours today. You can see if that works. So what we're going to do is, again, if you don't have a paper towel still or you need one, just raise your hand. But otherwise, we're going to take our slide, and I'll just talk through this so you'll be able to see. So first of all, make a note that it's still covered. If for some reason all your primary slid off, then you would want to note that, because that would be an issue. And the time, it was 1 hour and 25 minutes in primary. So then you're going to take your slide, you'll dip, actually we can do this part all together, so I'll kind of go slow. So you're going to pick your slide up out of your dish, you're going to tip it off onto your paper towel, just tip off that primary. Put it back in your dish and pour in some of your 1X PBS in there. So you take your slide out of the dish, you tip your primary off, yeah, just kind of shake it off on the paper towel, put your slide back in the dish, pour PBS in there, and then, yeah, everyone do that for me. I'll catch up.
That's kind of cool.
I'm going to need your blood transfer. Okay. So then once you've done one PBS, we're going to carefully hold your slide, pour it off into your liquid waste, pour some more PBS on there. So you're going to do 2 pour-offs, and then we're going to let it sit in the wash for about 5 minutes. Normally you wash a lot, so you might do 2 5-minute washes. So go ahead and dump PBS twice, and then pour PBS on a 3rd time, or TBS, and then just let your dish sit there. And then Darnell, before he left, asked a really great question about secondary antibodies, so I wanted to say a little bit more while we're just letting these soak. So what that wash is doing is any primary antibody that did not bind to our lamp 1, it's just washing that off. So Darnell had asked, like, for more clarification on why we don't just put our fluorophore on our primary. And I had said part of it was because it's cheaper to have a secondary against a lot of primaries. But there's a couple other reasons. You have more versatility. Like, there's multiple ways that you might want to detect with your secondary. So we're using a red, it's going to fluoresce red. That's because we're going to use a membrane stain that's green, so we can't use green. And we're going to use a stain for our nucleus that is blue, so we can't use blue. So we're going to use red. But we could use a green dye. Like maybe we only have a way to look at green dye. So if that was the case, then I can use a secondary that's anti-rabbit, but it's conjugated to a red dye. Or maybe I don't even want to use fluorescence. Maybe I want to use a different detection method. There's ways that you can detect that aren't using fluorescence, where you get like an image that you can look at in bright field, just regular light, and you get like a color substrate. In that case, it wouldn't even be a fluorophore, it would be a biotin or an HRP. You don't need to know what those things are right now. But basically, there's a lot of different ways you might detect for a Western blot, because we can use these antibodies for other things besides our cell staining. So if we were staining for a Western blot, we might use a chemiluminescence detection method. So that's going to be a different type of secondary. So that means we don't have to make our anti-rabbit primary, our anti-lamp primary, like we don't have to make 10 different versions of it. We can make one version of the anti-lamp, and then we can detect it however is appropriate with our secondary. So that helps. And then just another point, I've said this several times throughout, like in all of these assets, we really want like a strong contrast between our background noise and our signal, so that the secondary also can help amplify the signal. So your primary antibody, especially if it's a monoclonal, it will only bind to one spot on the protein. You'll have a very dim signal if you're only looking at the primary antibody, because it might only stick to a small part of the protein. But using your secondary, you can decorate, like, because actually more than one secondary might stick there too. You can amplify and decorate more of your protein and kind of build up that site, so you get a stronger signal, if that makes some sense. So this has probably been about 5 minutes in our wash now. So now we're going to put secondary on. So that's going to be the black tube. So what we're going to do is, again, pour off, and I'll just demo this really quick. I'm holding tightly. I'm going to just kind of whack my slide a couple times on the paper towel or my dish, so I shake off the excess liquid. This is all we have for secondary, so please use it sparingly. You're going to use your transfer pipette. You pipette up and down a couple times. Careful not to push it all out of the tube. And then remember, I'm keeping pressure so that my liquid's all the way at the bottom. And it should only take, I don't know, like 3, 4, 5 drops. And then I can use my, carefully use my tip to spread that liquid out into the square. So there should be enough for everyone, like at least 3 or 4 people to use one tube, but don't take it all out until your friends have used it. And if you need a little extra at the end, you can drop it on, if that makes sense. So you're gonna dump out your wash, whack your slides, whack your dish so your slide gets dry, and then transfer secondary onto there. And then once you get that secondary on, it's light sensitive, so you're gonna cover your dish with the foil.
Is your tube supposed to not have nothing because it's a little bit of liquid?
It's okay if there's a little bit of liquid still on there. Let's get it as dry as we can. So keep, once you draw it out, keep pressure on the bowl. Don't release the bowl. Secondary on. Put your foil on. And you notice that secondary is stored in a black tube. So even in its storage, it's protected from the light. Everybody have a secondary?
Suspended.
Oh,
You have 2 coils.
secondary? There's all of it? Okay. Does everyone use secondary over here? Is someone use some?
2.
He has one. And I have one.
And then, DAPI is blue. How am I gonna see blue? What's lower than violet? We can't see it, but a camera can. Ultraviolet, right? So ultraviolet or UV light, if we hit it with UV light and we have that cube, what color are we gonna get out? Just shift it a little down, blue. We're gonna get blue out. So, so our UV, I don't have it. UV, the excitation light will be UV, we'll get blue. Emissions, and that's going to be our DAPI stain, which is going to be our nucleus, and that's going to be the very last thing we stain. I always spell this word wrong. I'm just going to leave it like that. Someone wants to yell it out. So our red's going to be our high. So our blue light, when we hit with the blue cube, what are we going to get out? The next one up on the rainbow. I already said green. And this is going to be, we're going to stain the membrane with a WGA stain. And I'm going to put in parentheses here, it's GFP. Green fluorescent protein. And that's going to be our membrane. And then the last one, if we hit our sample with green light, what are we going to get out? Red. And that's what we're staining for right now. So that's going to be everywhere our lamp 1 signal is. So I'm going to write 594. Other common red dyes are Texas red and M cherry. We'll see those a lot. And this is going to be what? What are we looking at there? What was our primary against? It's on the sheet. I can't hear, a little louder. Begins with an L. What are we? Lamp 1. Exactly. So that's going to be our lamp 1 stain, which is going to be on our lysosomes. So we'll be able to see all of those with our hopefully, our microscopes. So, and then what's beyond red? Infrared, yeah. So there's dyes also that like the camera can collect that are looking in like the infrared region. So you can hit with red and get infrared. So that camera I showed you guys in the really small room in our lab that had like the beam splitter and there were all these crazy mirrors, that could actually hit your sample and filter out light of 4 different fluorescent channels and not get bleed through, and you can see them all at once. That's like, it was a crazy visit. It was one person's whole PhD to figure out how to do that. Ours is very simple. This is called a DAPI-FITC or wide field fluorescent microscope. So we're just going to hit it generally with the appropriate color and then collect the information. So, are there any questions on that? I know it's kind of complicated. No? Okay, that's good. That's great. Let me just look at how long our timer is going. Okay. Great. So we have time. Let's actually, I want to give you an idea of what we're going to see. So I just printed the data sheet, but in the middle of each bench are some sample pictures. So if you guys kind of gather around the ends of your bench, I have got one too. So when you go on these websites and look at these antibodies, if it's an antibody that's well used, you'll see some different types of data of people that, lots of times a company, if it's got a new antibody, you might be able to make a deal with them where you get it for free and test it out if you send your pictures at the end. So on this first page, for those of you that went into my lab with me last Saturday and saw the images up on the big screen by the microscope, we were looking at this. This is immunohistochemistry. So this is our LAMP1 antibody that we're using. and it's stained instead of, that's not fluorescent stain. So the secondary here is conjugated with something called HRP, and that allows us to get a reaction where we get a brown product around where our stain is. And these aren't cells, these are cells in tissue on this one. So that's what you're looking at there. And you'll notice on the side, so this was in formalin fixed, so that's that cross-linking, paraffin embedded, human kidney tissue. So it works there. We've got some other samples, but if you turn to this page, that third page, so you guys should have one. Oh, perfect. So this is page 5, it says at the bottom. So we're going to see the opposite of this. But this is, the nuclei in the middle is blue. And the lamp 1, it tells us, is stained with Alexafluor 488. So 488 is a green emission. So we stain with a red one. So all these dots, we're going to see them as red, not green. But our picture will hopefully look similar to this. And then the red, they used WGA, they used 594, the red for WGA. We're going to use GFP or 488, the green. But I wanted to point this out because this is a HeLa cell. Has anyone ever heard of HeLa cells? Yeah? A couple of us? Has anyone ever heard the name Henrietta Lacks or a book called The Immortal Life of Henrietta Lacks? Yeah? That's more familiar. So HeLa cells, they were taken from a woman at Johns Hopkins. She had a crazy aggressive form of cervical cancer. And they took some of her cells, and this was in the 50s, and we weren't able to grow cells outside of a body at that time. But they were taking samples from people that came in with cancer. The cancer cells grow well, and they were able to get these cells to grow. And they're the first cells that were ever cultured. And the story, I highly recommend this book, The Immortal Life of Henrietta Lacks. It's great science writing, but it's also like a very interesting cultural history. She was a black woman, she was a poor woman. The tissue was taken without her permission, and her family never knew. So these cells went out into the world, and all these labs, academic labs and commercial labs, studied and still study these cells. I've worked with HeLa cells, and their genomes have been sequenced, and we know everything about it. And her DNA is also her children's DNA. And this DNA got published in papers, and people had access to it without the family ever having any idea. So the family was very much taken advantage of, and in the process of sort of writing this and reading this story, not only do you learn how we were able to culture cells, which is an amazing scientific accomplishment, but it's a wonderful story of how medical ethics has evolved. And we now have to get permission, people have to sign off to have their tissue taken. There's a lot of questions, obviously, you know, with like these ancestry websites now and stuff, like it can be very dangerous for your DNA for someone to be able to get your DNA. Like there's a lot of ethical questions about that now. Should that information be private? It's a very interesting cultural and ethical history tied up into the story of how we got these HeLa cells. But almost every antibody you'll see a sample of a HeLa cell because they stain for everything. So it's very interesting, and I definitely recommend that book. But we will hopefully see this. But the last thing I wanted you to see, if we turn to the next page, page 7, this is also lamp 1 staining, but it's a different pattern. And there's a note here, so it's not dots. You see, so here the lamp 1 is, let me just make sure the dye is right, Alexafluor 488. Which again is green. But that's not dots. And the HeLa cells were dots. And they have this note here, and it says the filamentous pattern observed only occurs when cells are fixed with 100% methanol. How do we fix our cells? 100% methanol, right? And it says we are aware the staining pattern can be changed from cells fixed with 4% PFA. So we didn't do that type of staining. So we expect the dot staining, but you have to fix with PFA to get that. Because PFA keeps the morphology. So I don't know if we're going to see this filamentous staining, but when you treat with the alcohol, it says that lamp 1 is a lysosome marker, but also plays a role in cellular adhesion. So you see LAMP1 and then tubulin, which are, it's part of the cytoskeleton when you fix with methanol. So I don't know if we're gonna see that. Hopefully we will. But that's another important reason why you wanna read these sheets and understand your antibody. Because if someone told you, I want a lysosome stain, and you did this procedure, and then you went to the person and were like, why does it look like this? This is long lines, these aren't lysosomes. You want to understand why. And so in this case, it's the fixation method. So you need to make sure that the steps you're taking match the end result you want. I just wanted to show that because you can get very different looking results depending on what you've done in your procedure. And I think something we really wanted to communicate to you guys over the course of this wet lab is, yes, it's important to have good hands and learn these technical skills, but it's important to always be using your brain and understand what these different steps are, because that's going to ultimately help you interpret your results and become a scientist. And what I'm so proud of is when I brought you guys into SAM today, and I'm seeing it when we're walking around too, you guys are asking such good questions. And that's like the #1 important thing to be a good scientist. Like, you can learn how to pipette, you can learn how to do a fancy assay, or use a hard piece of equipment, but it's hard to teach someone to be curious and ask good questions. And you guys are all, I can tell you're thinking, and you're asking Sam, like, well, what if, is it good? Is it actually a good thing to have really strong bones? Like, can bones be too strong? Or like, I heard really good questions. Or what is this I'm seeing? Or why aren't all the cells on the dish? And thinking like that as you're doing science is essential. And that's what makes science actually fun, because then you're like solving a puzzle, and you're a detective, and you're not just like a robot doing pipetting. Which I don't think is what you want to do if you're interested in science. You want to solve puzzles. So, yes. Are there any questions about any of this? Good. So you guys can look at these sheets, and once we get to our images, we'll see if our pictures actually look like this. So after you make your, into your tube, after you make your secondary, we'll wrap it in a little foil. But this is going to be the small tubes. Where did Kyle go? Alright, so we're back to our benches, so we're going to have one each bench. One on each side. Hopefully this is one on each side. Great. I'm going to give you guys I'm going to borrow one of yours. Actually, we can have a third one. There's enough for a third one. So hopefully, I'm hoping this is enough. So don't mix this up with your secondary tube. Actually, your secondary tubes, the big black tubes, I will collect those. So I'm going to return that big black tube. So again, the WGA was previously aliquoted because you shouldn't freeze thaw it a whole bunch of times. Big black tube. Good. Did you guys use up all the big black tube? Oh, perfect.
Oh, we only have one.
Yeah, you might have only had one. Perfect. Okay. So, you need a new orange tube. Does everyone have a fresh orange tube? Okay. So take a fresh one, pass them around. And it's going to be the exact same as before. So we're doing, you can write WGA on the top so you don't confuse yourself. I would actually recommend taking your primary at this point and just throwing it in your, you can put it in your Sharps waste thing. Just so you don't mix it up and accidentally put primary back on. And so I'm doing a we're going to use the antibody dilute we made before. We're doing a 1 to 200 dilution. I'm going to make 1 ml of it. So how much of my diluent do I need? 995. That's right. Whoops. Just poured it down. No problem. Thank you. We're going to be okay. And then into the Sharps. And then how much of my WGA do I need? 5. Yeah, and I need to borrow one of your guys's. So I'm going to do 5 of, I need to borrow the little black tube. Yeah. I'm hoping this is all spun down to the bottom. If it's not, if you can't get at it, let me know. And then I'm going to mix it, and then what I'm going to do after I mix it is I'm just going to break off a little piece of foil and just wrap my tube in foil. So everyone can start doing that if you're comfortable. These tubes are a little dark and it's not going to be in the light for that long. It's more for the video. I hope so.
I'm not playing games today. Not today. I'm not. You beat that.
Don't cover the top. You have to open it and step out. You only need a little. You only need like a little. What was that? No, but there is another one on the table. It takes the they might have it here. It takes these tips. These are the ones on the table.
Try again. Have the button pressed down and slowly release it.
Interesting.
So I'm letting that timer go on
No, I think you
purpose
have something. There's like
to
a
train you
video.
guys not to panic when you hear the timers going off. You'll hear a timer go off all the time. It stops after a minute. So we're gonna immediately be washing and doing this.
Let's try to stay up there. I see.
So once you're ready, again, this is the same technique we've been doing. So we're going to and this is another reason why you don't want to tuck under the lid. So see, I can just like lift this now and I have access to my slide. I make sure that it's still covered, and it is. I'm just going to lift my slide out, tip off the secondary, put it back in. I'll do 2 rinses, just like we've done before, and dump, and then the 3rd rinse, I'm going to leave on there until we've all caught up with each other. 2 rinses, and then leave it, put it a 3rd time, put your PBS on, and leave it there until we're all caught up. When it's sitting there in PBS, cover it again with your foil lid. And if you're running low, I've got some more. Like if you guys were not sleeping.
Yeah, we're probably gonna need more PBS.
You need a little more? Okay. I've got, oh, actually, you know what? You can use your diluent. We don't need it anymore. The PBS-T, you can use that for the wash too. It will be fine. So if you need a little bit more, use the PBS-T that you made. Okay. It's not gross. That's okay.
So do we cover it after?
We need some washes over here. Okay, get that washed up. So I want to do the WGA all at once. It's going to be the exact same thing. We're going to dump out all the PBS. We're going to keep our hand on the side and like shake it on our paper towel. You can use all your WGA. You're just going to fill up that square with WGA, and I'm going to start the timer for 10 minutes.
Start right now?
So we'll start now. So you pour off your PBS. Just tap your slide dry. And then you're going to take a transfer pipette, and you don't have to save the WGA, so just fill up that square in your pack.
Smashes it on.
And then make sure you cover your dish, and then I'm protect it from light. When you're pipetting, keep your dish on the table, because lifting it up and down has more chance for spilling. Sure. I put them in there. All right. So everybody should be like covered. So we're gonna be imaging soon. It depends on the type of lamps, but a lot of times lamps need to be warmed up. So we're gonna warm up our lamp. I think this one already on. I think this one's already on. So there's a switch at the bottom of the lamp. That's the square on the side. You're gonna push that switch up. So you're gonna turn it on. The wheel makes the light more or less intense. Usually, especially if it's a laser that's emitting the light, you don't want to turn those on and off and on and off. You'll wear out the lamps, and they're very expensive to fix. So we're just going to put that on. It's going to take like 10, 20 minutes to warm up, I think. Don't worry about that cord. Don't worry, it doesn't go to everything. Yeah. So you just make sure your lamp's on. You'll know it's on because in like 5 minutes or so we'll get warm. This one is warm. I have very little cover slip in medium, so we're going to do it like in a controlled fashion in groups. So all of us can, at the same time, we're going to dump our excess WGA off, tap your slide, 2 rinses with your PBS. And then you're gonna let it sit in PBS covered, and then you guys are gonna make your way over here, and I'm gonna show you the cover slip. Oh, it's a dropper. Oh, it's tricky because here bubbles matter. So if there's a big bubble in your sample, you're going to affect your imaging. So there's different ways you cover slip, but I have the best way. I learned it from the very first grad student I ever worked with, and whenever I've shown any other lab this, or person that's cover slip forever, they're like enthralled. So the cover glass is very, very thin. So we'll do this half now. The rest of you can just hang out at the bench and then we'll swap it. The cover glass, so you kind of get used, you want to just hold it on the edge. You don't want a big fingerprint in the middle. And you kind of get used to, if you bend it a little bit, what it feels like when there's one and when there's 2 or 3 stuck together. And that's just practice. But there is a trick. If there's 2 stuck together and you hold it up to the light, you'll see a rainbow, kind of like rain on a window. So then you know you have 2. But that takes some practice. So I put a little bit of gauze, a soft pad. You can also fold up some paper towels. But I put gauze in everyone's. So I'm going to put my cover glass down here. And then I've got a fake slide here. Right, thank you. So what I'll do is, this is fine. And I need Kim wipes. Do you mind describing the Kim wipes? Yeah, or a couple of them is fine. Yeah. So take a Kim wipe. So I'm going to take, this is the mounting media with DAPI. This happens to come in a dropper bottle. So the mounting medium helps prevent photo bleaching. There's only so many times you can excite an electron and get a photon. After a while you get something called photo bleaching and that area will just be black. You won't have color anymore. So you want to, there's stuff in this medium to help prevent that and fading to an extent. But I'm going to just tap that. I'm going to dry the back of this, and I'm going to dry the part that's not my pap pin, so I don't want to dry my cells. And then what you're going to do is you're going to, and this we want to be kind of conservative with, you'll drop a little bit of this DAPI, so our stain for our nuclei is in here. It's just a little bit, like a drop. And then you're going, my cover slip's down here on the soft pad. I'm going to turn this away from me. I'm about 1/3 of the way up on the side, and I push down and into that pad and slowly tip my my slide towards my cover slip. And what happens is the liquid draws up nice and smooth, and I squeeze out any bubbles, and then like this. And then I have my cover slip on there nice and smooth. I got a couple of bubbles right in that corner, but it's not in the middle where it matters. If it is where it matters, you can sometimes kind of gently push or push it more. But that's a nice smooth, like some people try to drop it on and you'll see a lot of different ways. this technique will impress everyone. So I have only 2 of these, and this is, we don't have a lot. So what I'll do is I'll go to, stations. But I just want to demo this for the other half. It doesn't hurt. The WGA statement's pretty strong. So I want you guys to go back to your slides now, do one more wash, and just let it sit there, and then I'll come around. It won't hurt them to wash. Yep, so you can dump your slide, and then I'm putting a very small drop just in my square. And then I push gently against the edge, and I just let that liquid fill up my square, basically.
This is gonna take a while.
Huh?
It's gonna take a while for us to get.
I know, but we'll be good. And then when I'm done, I'm gonna cover my slide. Remind me my slide's here when I'm looking for it. Just wipe it. All right, so hold it flat with one hand.
Okay.
Drop in the middle of your square. Drop a line. Yep, you guys can do it too. So you got the back to my side. Yeah. Right on the edge. Good. All right, put the dropper back in. Now take your slide. Hold it like this in 2 hands. No cover slips on the ground. Yep. You're gonna tilt away from you and gently push the edge on your cover slip. This is the hardest part of the whole. So put it a third of the way up.
Okay.
Tilt it away from you. Don't push too hard. Away from you. Yep, it's gonna start to drip on the slide. Keep tilting away, keep going, keep going. Gently draw the slide towards you so you catch the edge. Good. And then see as you start to push, bring this up and it pulls the lid up. It pulls the slip on. There you go. And now push really hard straight up and down. And then flip it 180 degrees and push really hard straight up and down. Yep. There you go. And your cover slipped, and you're just gonna cover it with foil and set it down. Yep. No.
Just cover it?
Cover it in foil. Now you've got a slip. Yep. Yep. And we'll image it shortly.
Perfect.
Okay, perfect. So now we're gonna look at these. Did it work? What do you guys think? This is the true feeling of a scientist. At the end of the day, and you've done a long experiment, and you're like, please work. So, all right, microscope's on. There's a switch at the bottom right-hand side of your microscope. Camera's on. There is a switch on the top left of your camera.
Yeah.
Top left of the camera, turn it on. Okay, so we've got our camera on. We're going to analyze. Where did I end up putting my slide? I had a slide. So let's gather around here. We're gonna dim the lights as we're doing this, but we'll start it. I just want to show you a couple components. So your cube is at the bottom. So you see a G, a B, and a U. U is for UV, B is for blue light, G is for green light. You'll notice, actually, can we turn off the lights? They're right by the phone. What color is coming out of my microscope? Green. What color is on the camera? Right. So that's our excitation, our emission. So down here, if I slide the cube in one, what color is coming out of my microscope? And what color is this? Green. Greenish, yeah, green. And then finally, if I push this cube one more. Ultraviolet? Yeah, it's ultraviolet, and we'll see blue in the, and yeah, it's white light. Oops, our camera turned off. Okay, did I just take a photo? Okay, so let's see if, so this should do, this just looks like, there we go, okay. So, so if you have the lamp on, that's the, dial at the bottom, that's bright field. If I turn it off now, I'm in my UV. And we do actually have, so that's just a speck, but let's see if we can find, so it's easiest to find, I'm gonna actually, and you guys learned about this, you have eyepiece all the way out as camera, in the middle is both. So let me see if I can find DAPI in my eyepiece. Okay, so DAPI, oh goodness. DAPI is always the easiest to find. Your cell, like that state is impossible to mess up. So I'm putting this back in camera. So there's our cell nuclei. So it doesn't look super great, the WGA, but so if I push it to green, I'm gonna have to, we can take up the intensity, there we go. The intensity of our stain, and so there's our green. Our green channel, so that's our WGA. I'm gonna zoom in on it so we can. But there's our cell membrane, and then I don't know if we're gonna have a red signal, but let's see. I'm gonna take up the red one. I'm gonna be honest. It's a very old antibody, so it may have not worked. So I'm gonna focus that, so that's a little closer. So there's our cell nuclei. Yeah, they're pretty far apart. And then if I push it one, there's our cell membrane. Do you see how we're getting the whole shape of the cell there? So if I push it back one, it's just the center of the cells. That's our nuclei stain. There's our green stain. And then, I don't know. Oh! It's very faint, but you can see, so we'll be able to amplify that, but there's like some red dots in here. So what we're gonna do is we're gonna take a picture in each channel, and then we're gonna overlay them, and we'll be able to get an image like they have in there. So I'm gonna actually zoom in one more, because then we can see it more zoomed in. So I found an area, I found myself like blue. Okay. And I can balance out that, yeah. So now I'm gonna go back to green. And so I see how it's dim? So I'm taking the intensity on my lamp up until the green's nice and bright. And you can adjust the fine focus a little bit. So that's our cell membrane stain. Oops, that overexposed it a little bit when I took the picture, so I'm gonna, wow, it takes a really bright picture, so I'm gonna actually dim it. So there's our green. And then I'm gonna push it to red. And I haven't moved my slide, and let's see what happens if I take that picture. Sorry, I'm good. Oh, there's a red. And it looks a lot like the green stain, right? So remember we fixed in methanol, and then we stained the cytoskeleton as well. So it's gonna overlay. Okay? So you guys, Kyle and I will be around to help. You guys can look at some of your slides and take some pictures. I think it's about 4, so we'll take around 30 minutes, and then we'll throw one of them up on a computer, and I'll show you how to overlay it, and then we'll send it out. So, okay? And we'll be around to help. It takes a little bit of fussing to figure out, but just practice. Okay, so it's easiest to find this. So this is the bright side. It's easiest to find DAPI. So I'm pushing the cube down here. I'm sliding it all the way to. So that's our ultraviolet line. Yeah. So I'm sliding it. So you see the dot under the U. That tells you that you're looking at the U cube. So if you move it one. Wait, there's another one. Keep moving till you feel it click, and then. Okay, so. Yeah, so here is U, the middle is B, you just have to remember it. And then if I push it all the way this way, so just wait for the light to G. But you'll also see the light, yeah. So let's start with UV because it's easiest to find cell nuclei. Whose slide is it? The camera right now doesn't have any feed because this is closed. So this is shuttered so that you only have the eyepiece. Do you see the side here? Yeah. In between is both. All the way out is, just camera. I thought we were done with it. So in the middle, and I find it easier to find things by eye. So that's because we have, I don't know why yours is.
Whose slide is this? I think it was an example one that.
Wait, it was an example one?
This was taken from
Here?
Yeah.
Oh, then.
Oh, okay. But you have a lot of dye spill though. So I don't. Oh, there we go. There we go. So the reason why yours, I think, is purple on the back is remember how you have that dye spill on the back, all that DAPI. So I think that's probably its imaging for the DAPI.
So we're supposed to be getting.
Oh, it's supposed to be black.
It should be black in the back. We can fix that in image J. But what happened is when your slide was being cover slipped, stain spilled on the back, and so we're looking for that. So actually, I think we should look through a different slide. Yeah. But you've got stain, and we can actually. So I don't know what that's going to do for the green channel.
Kind of like.
Yeah, green looks great,
We just need to be zoom.
and then red. Yeah, so it's easier to zoom. So find it like it's easier. So it's easier to find things wide and on blue. And what you're gonna look for is you're gonna look for an area where you see a lot of like blue cells. But yeah, let's just use a different slide just because I think that stain is yeah great. Oh, so then if you see it with two slides and you know nothing happened to that other slide, then I would say that it's probably the lens is filthy. So I'm gonna just take a little bit. I'm gonna just, so you actually should use lens paper. I don't have lens paper, but this is so dirty that and I can see it's dirty. I'm just gonna get, so I bet something spilled on this. Okay, so I would find an area where you have like this, oops, yeah, you can move it, I'll let you find it. But like, if you have a clump of cells together, it'll be cool because you'll see them attached to each other. Yeah, because you're gonna zoom in. Like, go where there's a clump of cells. No, that's one. So go like there. Yeah, all right. Like put that right in the middle. That's okay, that's okay. Anywhere in here is fine. Okay.
Oh my gosh.
I think.
Okay, up and down, right?
Yeah, so that's.
Oh yeah.
Yeah,
that's why.
this is weird. Yeah, it could be easier than I
Towards
just
it.
would.
Oh my gosh.
But it doesn't. It's not totally matter. Like set it down. Stop. Okay, perfect. There it is. So to go to a higher objective, a higher magnification, you spin the bottom here, the wheel, and that's what. So it's our lens. So you see how now that red went away.
It's just that one objective.
It's the objective is dirty. Yeah, so you can kind of move that around. Yeah. So, oops, I see the trouble. But so anyway, so then you find an area that's good, and you can even try like one more if you want. So I'm gonna just spin down here. That's just gonna be a couple cells if you want to get it zoomed in. So then you focus it with the small knob. And then I'm just going to take down, and so I'm taking down the lamp intensity. Teacher's going to be brighter, but also DAPI is so bright that you don't need to hit it with the full power of your knife. So there. So if you want to like try that, you can also see preview what it's going to look like if you want to slide the cube. Yep. So there you go. So you don't want to move the slide now, right? Because you're going to want to take the picture in the same spot. But you might want to take the brightness up. And then, we have some red signal too. But for red, especially, you want to decrease it. And you might have to adjust the focus before you take that picture. Because the nucleus and the cell membrane, it might be in a slightly different plane. So just get it so focus. And then to take the picture, it's this button here. It's okay if the camera moves, just don't move your slide once you find a spot you like. And you can mess around, like if you take, I'm gonna take it as a preview. If it like blows it out like that, we can fix some of that in image A, but then maybe you want to take your lamp. Like take it brighter to find it, but then take it down. Take the picture and then see. Yeah, and then I'll show you a way if we have time that we can adjust those pictures. So, but then once you find a spot you're happy, then you take the same picture in all 3 channels. Does that all make sense? Okay. So you guys can play around with your slides. And also while you're doing that, you guys can also chuck basically everything on your slide. So let's actually take a pause for a minute, and I'm going to show you guys on my laptop. We can plug the computers into the camera, I'm just going to show you an idea of then what we would do. So, I plugged the SD card in. I'm going to take a chair. And I'm going to open our 3 images in a program called ImageJ. Fuji is also ImageJ. It's a really common free image software, and it's really easy to manipulate images. So I'm opening that now. You can download it for free. Remind me later. Okay. So here's the blue channel, and I'm going to take the green channel, and this is why it's important to not move your camera, and then I'll take that red one. Looks good. And I should be able to, so the first thing I'm going to do is I'm going to, sorry, those open to preview actually. Let me drag these. So I'm going to drag this into ImageJ. Perfect. Oops, this one. So, and a lot of times, in labs you'll see like microscopes are like attached to computers, and there's programs. Like you guys saw that in our lab, the little room. So you'll have a special program, and you can do a lot of this within the program. So you would be able to but you'd be able to adjust your lighting. We're doing it manually with the intensity, but you would be able to adjust, like, so you're only exposing for like 100 seconds, and you'd adjust your laser power and all this stuff. So what I'm going to do is I'm going to take this image first. I'm going to adjust the brightness contrast on the red, and basically I'm just here. Let's see what happens if we do auto. No, that's reset. So I'm going to try to take out the lower level red light, which is this background, and this is a little bit because your lighting was off, so it's more exposed on one side, so it's like nicer there. And then so I'm just adjusting my maximum and my minimum values that it's seeing, so that I'm capturing just the actual red signal. And because of the way the picture was taken, I'm not going to be able to do it perfectly, but you can see, like, I'm taking out just like red that's in the background, hopefully, and keeping the most intense stain. So I'm going to do that for the best I can for each of mine. So now I'm going to adjust this one, process, image adjust, brightness contrast. So same thing with the green. So I want to cut out the minimum, like the lowest values, because that's just background. But I want to keep my most intense values. So this is just kind of grossly adjusting this. And I'm going to have I have the same kind of overexposure. There might also be a little bubble right there. That's what happens sometimes with bubbles, is the light, but we'll see. Okay. And then DAPI usually takes very minimal because it's so bright. So I'm going to take out the background, and then I have my new guy apply. And then what I can do is I can stack these images. So let's see if I can remember how to do this. Colors. Let's see. Stack. Make a composite. I think that's what I do with this. Oh, that took forever to open up. Okay. Oh, sorry. So we got our image numbers up here. So let's do image. Merge. Okay, so red is going to be 36. Green is 34. And blue is that might be. Let's see what happens. Ah, there we go. So there's your second. So what I would do is I would try to take a picture on the camera, like pick an air, like so like that looks cool, but where you're not getting that overexposure.
At 40x.
And do it at 40x. And I think you guys, yeah, on any of your slides. But so then you see you have your 3 channels. So then you can go back and you can like adjust the green and the red. Yeah, nice. And then, do you know why it's yellow? Like when we merge it? Because the colors. there's so much overlap of the red and the green because we've stained the same elements basically, which we expect with the methanol staining. So this is good. Like this group, you can see they did a really nice job focusing our here. So we should get a good composite. And this is nice because you see these cells. Remember, these are the bone cells and they're dendritic. They have all those arms. So we have some really nice arms here. And I said it to the group when I was over with this group here. But do you see all this staining in the middle of the cell near the nucleus? The nucleus? Does anybody have an idea that I didn't already tell what that is? Like what's next to the nucleus? Not the cytoplasm. It's covered in membrane. The endoplasmic reticulum. So that's actually, because our cell was permeabilized, we were able to get our staining in really well. So we're also staining the membrane inside of the cell as well. So that's what we're seeing. And then, actually, I really like this red image because we do have some staining in the membrane, but we also do see where we have these big dots that are stained, these big circles. Those probably are lysosomes. So it's totally legit. So I'm just going to go through and adjust. So I'm taking out the lowest red values. I'm basically increasing the contrast. I'm increasing the contrast. So I'm cutting out the like really low level red that's just the background. Sorry, I know it's kind of hard to see. The reason why we have quite a bit of background is because we were a little lazy on our washes. We were doing this for like, we had to take a picture to put in a magazine. We would wash a lot between each step. So we don't quite wash it. But anyway, it's fine. So we're going to apply that. We're going to do this to the green really quick. This green is beautiful. Yeah, so you would wash off the extra stain and just like get it out of there. Because the stain can stick, sometimes like it can stick to the slide a little bit, it can stick to other parts of the tissue. So like this stain that's like part of it is the exposure, part of it is we just didn't wash. So normally when I'm washing for staining, I do like 3 5-minute washes. But it's fine. Ooh, that looks good. Nice. I'm gonna see if I can get that one really good. There. Okay, and then the last one. I will say you always have to be very careful when you're adjusting images. These are qualitative pictures. We're not using these to make any scientific claim other than that these structures are in there. Like it is possible to adjust an image so you adjust one so it looks like there's a lot of signal and other ones that so you never ever do that if it's data and it's a qualitative picture. You have to be very careful on that. But we're making a pretty picture. So then I'm just gonna merge this. Our blue channel is 7656. Our green channel is 7654. And the other tricky thing is, remember, a lot of times fluorescent cameras are actually black and white, and we pseudo-color them. So you always have to be aware of which picture is which when you're merging your channels. This must be 51. Okay. So here you go. And there's our cell. Very nice. So, and it's yellow because there's a lot of overlap between the green and the red staining. So I'll make a nice slide for you guys. But like last times what you would show is like each channel and then your composite image. So the yellow is where red and green are co-localized. So, but yeah, you guys, you're awesome. Do you feel good? Do you guys feel like you've learned a lot in the wet lab? Yeah? So we have like I think about 20 minutes, which we don't have to fill. If you want your slide, you can. I guess we can't really look at it otherwise. We had a really good question. So what do you do with these slides? If you store them in the fridge at 4 degrees, you could usually get an image. They'll be good for 2 to 3 weeks. You protect them from light. That mounting media helps protect them. But over time, you're going to lose that fluorescence. So if you are doing fluorescent staining, you always want to get your images as soon as you can. It would be okay if like it's the end of the day and you can't take pictures until tomorrow, where there's some stuff's come up and you'll get pictures on Thursday. But you want to be aware, like you do all that work, you don't want to like leave it in the fridge for a month and then go get it and have no stain anymore. So yeah. But were there any questions about like any of that stuff or what we did? Or, I do want to say, like, the internet is incredibly helpful, and use that, and don't be embarrassed to Google things or start with Wikipedia things. I did want to, maybe I'll send an email out, but so where the antibody came from, AbCam, they have webinars, so there's a lot of, like, this assays up there. There's a lot of, like, if you're being told, like, oh, you're going to do IHC, or oh, you're going to do, like there's a lot of different things you might do. A Western blot, like you can go on AbCam, they have videos, and they explain what all the steps are doing. So that can be really helpful, because some grad students are good at explaining some things and some aren't. So it's always good to have those resources. YouTube is amazing. It's I can't believe the stuff people take time to put up on YouTube. There'll be whole lectures. There'll be people like drawing out diagrams of signaling pathways. It's great. And for microscopes, there's a website called Microscopy U, the letter U. So they like explain like this type of stuff with the fluorescent channels and how they work. If there's a really fancy microscope in your lab, or a focal microscope, or a super resolution microscope, and the grad students are talking and you're like, what does that even mean? And you're too embarrassed to ask them, you can start somewhere like Microscopy U and read about how it works, and then ask them more detailed questions. But I use the internet all the time, and I want to tell you my favorite internet story. I was dissecting some mice about a month ago. And I've dissected a lot of mice, but I've never dissected pancreas. And so kind of last minute, I was told by my boss, like, oh yeah, like I'm supposed to take a spleen and a liver, like take the pancreas out too. Like, all right, yeah, fine. So I go down there and I start dissecting my mouse, and then I take out the spleen, and I take out the liver, and then I'm looking at it and I'm like, I'm not even really sure where the pancreas is. Like I have an idea of where it is, but I don't even know like which thing this is. So they have computers down there, and like with my mouse open up on the table, went and Googled like mouse pancreas dissection, and I found a video, and somebody very nicely explained on the video how to dissect out a pancreas from a mouse, and I did that like in the middle of my experiment to get that pancreas. So like definitely don't, I guess the lesson is like don't feel like you have to know everything before you go, and a lot of science is like improvising and adapting on your feet, and like you don't always have someone to ask. Like the internet's a great resource. If you have a grad student, another like intern, whoever, like they might know too. Like, but yeah, I was down there by myself. And I was like, what am I gonna do? Like I'm supposed to bring up 35 pancreas. I don't even know how to get it out. So YouTube definitely saved my bum. So yeah. So, but yeah, I really hope you guys have like, whether it's this summer or in the future, like maybe you don't get it up in a wet lab this summer, but I hope you guys get a chance to do a more extensive internship at some point at wet lab. You learn a lot, and I hope it's opened up your eyes to science kind of outside of just like reading about it in your textbook or doing an experiment with some food coloring. So, yeah. What was that? What was that?
Like the speech, it sounds like you're saying good luck.
Oh, and I'll never see you again. No, I was actually gonna say, I'm gonna write my email up too. Please write it down. It can be this summer, it can be in 4 years when you're graduating from college and you have some questions you wanna ask. It could be because you wanna know more about Regeneron. It could be you just wanna get a coffee and like talk science or talk about something. Like, I've had so much help in my life with people mentoring me through different stages and helping me make decisions. And I'm totally a pay-it-forward person, so I've been super impressed with you all. I will see you guys in another course, but this is it for the wet lab stuff for now. And like, please take down my email, and when you eventually, I don't think you're on LinkedIn yet, but someday you'll be looking for a job and get on LinkedIn, like find me. And keep me updated. You can always just email me too and say, in 2 years, hey, I did an ICC and they were really impressed because I knew what they were talking about, or whatever it is. Like, please think of me. Yeah, thank you very much. I want to give you guys all a big hand. I'm gonna drop my email up here.